Hello everyone. Welcome back to our continued discussion of the immune system. Today we'll be discussing Chapter 18, the adaptive or specific immunity. Adaptive immunity or specific immunity includes defenses that are fine-tuned to a particular pathogen and can be retained for future encounters. It has two parts. The humoral immunity occurs in extracellular fluids, and it acts against pathogens that haven't or have not yet infected host cells, and it is mediated by B cell activity and antibodies. The second part is cellular immunity, and this occurs on infected host cells, which is mediated by T cell or T lymphocyte activity. Humoral immunity uses antibodies to recognize antigens. Antibodies are proteins produced by white blood cells in order to recognize molecular signals such as polysaccharides or proteins present in foreign entities. These are called antigens. Antibodies are found on the surface of B cells or secreted by plasma cells. Antigens may have a variety of sources. They may be part of a pathogen's cell wall, the capsule, or viral coat. They may be the fimbriae or flagellum. They may be particular bacterial toxins. And antigens can also be non-pathogenic. So often when we develop allergies or we may have organ rejections during organ transplants, these are two examples of a body reacting to non-pathogenic antigens. Epitopes are the specific parts of an antigen that are recognized by the antibody. As you can see in this image to the right, you can see how antibodies bind to different epitopes of a particular antigen. This slide covers the structure and function of antibodies. There's a lot going on in this slide, so pay close attention to the figure on the right. Most human antibodies are bivalent. That means that they have two antigen binding sites, which are located at the tips of the fork of the antibody. They are called V for variable. So this is the region that varies in the antibody to be finely tuned to different types of antigens from specific pathogens. There are two identical light chains, so VL on both sides, and there are two identical heavy protein chains. The light and heavy refers to the molecular weight of the protein that comprises them. So that is the sort of the business end of the antibody that binds to the epitopes. Next we have the stem. The stem is called the effector constant or FC region. This area is constant and doesn't change necessarily between different memories to different types of epitopes. When scientists look at this region, they can classify the type of antibody based upon the effector region. The effector region is bound to the antigen binding sites through a short polypeptide with disulfide bonds. And that is all you need to be aware of about the image to the right. You should be able to draw that forked region and be able to efficient or clearly label the antigen binding sites, the variable regions, and the effector or the constant regions. You should also be aware that VL and VH are the variable light and heavy proteins, and the C is the constant stem region.
Checkpoint 1. Epitopes are the specific parts of N blank that are recognized by N blank. Checkpoint 2. Which answer best describes the regions of the antibody?
And now this table is a summary of the immunoglobulin classes. Remember, the classes can be identified by looking at the stem region. Each class has its own structure, the percentage found out of the total serum antibodies, the locations it can be found, the masses, how long it lasts in blood serum, its complement fixation. So remember, there were complement proteins that enhance the function of the immune system, whether or not it can be transferred through to the placenta, and its known functions. So different things that it can attack to and what it specializes in. Immunoglobulin is a class of proteins that is almost exclusively composed of antibodies. So for all intents and purposes, the terms can be used synonymously. There are 5 major classes that we need to be aware of. There are the IgGs, the IgMs, the IgAs, the IgDs, and the IgEs. It would be a good idea for you to print off this table and study this table in particular. Now let's break down how each one of these classes vary and what unique components we can see for each immunoglobulin class. Most of their structures are the same. Most of them exist as monomers. However, the IgM class can exist as a pentamer, and the IgA class can exist as a dimer. As a percentage of the total serum antibodies, the major immunoglobulin class is the IgG. The IgG is present in the blood, lymph, and intestines. The next highest percent total serum antibody is the IgA. The IgA is unique because it has a secretory component, so it can be found in secretions such as tears, saliva, mucus, the intestines, milk, and also found in the blood and lymph. Lastly, notice that IgE is always bound to mast and basophil cells. And recall that basophil cells have to do with histamine release. These are found throughout the body and blood. We can ignore the molecular mass for now, and let's look at the half-life in serum. Here you'll notice that IgGs have a very long half-life in serum. So after 23 days, a particular IgG with its own specific variable chains, only about half of them will decay after 30 days. Don't worry about complement fixation. Let's now move on to placental transfer, and here you'll see that the IgGs are capable of being transferred to the placenta, through the placenta. So as you can imagine, IgGs representing a high percentage of total serum antibody, having a long half-life, and being able to complete placental transfer, they have a very general function. They enhance phagocytosis, they neutralize toxins and viruses, and they protect the fetus and newborn babies. IgM is unique because it is typically the first antibody produced in response to an initial infection. IgAs are unique in that we discussed that they have the secretory element, so they can be localized for protection on mucosal membranes. And as we discussed, IgE is present and bound to mast and basophil cells that are responsible for histamine release. So you can imagine these are very important in our allergic reactions, but these cells are also very important in combating parasites. So it might be play an important role in the lysis of things such as parasitic worms. So there's a lot to process in this table, but what I just discussed are the important notes that you should be familiar with.
Checkpoint 3. Which antibody exists as a pentamer?
So now let's talk about the function of B cells in the immune system. First, we'll discuss the activation of B cells. Now, there is not just one B cell in the body. There are many different B cells in the body that come from the bone marrow, and they each have their own unique variable region on their antibody. They only produce one type of variable region on their antibody, but each B cell that's produced from the bone marrow has a unique antibody to itself that varies. If that B cell happens to win the antibody lottery, and as it's floating around in the body, it finds an antigen that matches its own special antibody, then it will make contact and bind to that antigen. Next, the antigen is engulfed and digested by the B cell. And that fragment of the digested antigen is then displayed on the outer cell surface. Next, that displayed antigen fragment attracts a T helper cell, which releases cytokines, and that activates the B cell. Once the B cell becomes activated, it divides into plasma cells. Remember, we said that B cells and plasma cells can produce antibodies. These plasma cells then produce much more of those antibodies. So that is activation. Now let's discuss clonal expansion. You see, if a B cell is quote unquote fortunate enough to find an antigen out there that matches its antibody, it will differentiate into a memory B cell that can rapidly recognize the same antigen at future encounters. So here we see at this graph or this image, we have a B cell, its antibody's variable region recognizes an epitope on an antigen. It can differentiate, therefore, into either memory B cells and plasma cells and make a monoclonal expansion. So many more cells of that particular B cell will be replicated or cloned. So here we can see how the adaptive immune system can be specific for an individual pathogen and also have a memory. Okay, so as we've discussed, the adaptive or specific immunity includes defenses that are fine-tuned to a particular pathogen and can be retained or have memory for future encounters. It has two parts. We've discussed the humoral immunity. The humoral immunity kills pathogens before they enter and infect host cells. Next, we are going to discuss the cellular immunity. The cellular immunity kills pathogens as they are inside the cells. So let's cover the classes of T cells. T cells are classified based upon their what are known as clusters of differentiation, or the sets of glycoproteins that mediate adhesion to receptors. T helper cells recognize those antigens that were displayed on the surfaces of phagocytes we discussed with B cells already. So you have your macrophages, your dendritic cells, etc. And then they produce cytokines that activate them. This doesn't always go well, so we also have something called T regulatory cells. And they destroy the helper T cells that cause autoimmune reactions, including against normal microbiota and against the fetus during pregnancy. Cytotoxic T lymphocytes recognize host cells that have been infected with a pathogen, and they release perforin and granzymes to help destroy them.
Checkpoint 4. Fill in the blanks. T blank cells recognize antigens displayed on the surface of phagocytes, macrophages, dendritic cells, etc., and produce blank that activate them.
Here is an article in which it discusses how the FDA approves the first gene therapy for leukemia. Leukemia is a cancer of the blood. And here the treatment involves removing T cells from a patient, purifying them, and then genetically altering them so that they have new variable regions associated with them that allow them to bind and identify cancerous elements. They are then tested for any sort of autoimmune reactions, and if they're okay and do not cause any issues with autoimmune disorders, they are re-injected into the body, and there they go through their own monoclonal development and expansion and seek out and destroy tumor cells. They are called CAR T cells, or chimeric antigen receptor T cells. Chimera because they're the combination of two different kinds of cells, and they include an antigen receptor that's added to the T cell. So now let's move our conversation from T cells to how immunological memory develops. In this image, we can see the time after initial exposure to a pathogen on the x-axis, and on the y-axis we can see the titer or concentration of an antibody in the serum. At about day 52, this figure demonstrates what happens if a second exposure happens with that particular pathogen. So let's first look at the initial exposure. As previously mentioned, the first antibody on the scene is IgM. So you see that IgM doing an initial peak, and then it is slowly replaced with IgG, which is the primary antibody in the blood serum. And therefore we see IgM drop down and IgG increase. The green line represents the total antibody titer. So you can see overall the total antibody titer increases. As the infection decreases, we see that the total antibody count goes down along with the IgGs. And the antibody production peaks in 2 days in secondary response versus 7 days in the primary response. So there's a lot faster response to a second exposure, and this is all part of the memory of the immune system. So those IgGs and some of those IgMs remain around in the bloodstream from a previous exposure, and so they're more capable to rapidly go through that monoclonal expansion or the release of those antibodies from a plasma cell that we discussed before. So let's break down adaptive immunity into different categories. It can be naturally acquired or artificially acquired. Naturally acquired adaptive immunity is one that you get from some sort of incidental exposure to an antigen or antibodies. And an artificially acquired adaptive immunity is one that you get from deliberate exposure to an antigen or antibodies through some sort of medical guidance. Let's look at naturally acquired first. So we can break it down into being active or passive. Active comes from direct exposure to an antigen. Remember that the antigen is hopefully a component of an invading pathogen. Passive can be when you just acquire antibodies from another individual. so you don't have to go through that whole process, the process in which antigens enter the body naturally and the body induces its own antibodies and specialized lymphocytes. Instead, with passive naturally acquired, antibodies would pass from, say, a mother to fetus via the placenta or to the infant via the mother's milk. If you look at the other side, we see artificially acquired can also be done through active or passive. Active would be a simple vaccination where antigens from a pathogen are injected into a patient, and then the patient goes through the process of producing its own antibodies and specialized lymphocytes. Passive artificially acquired is when the antibodies are injected into an individual or a patient, and this could be something like an anti-serum to a poison.
Checkpoint 5. Eli Lilly has developed antibody therapies for COVID-19. These therapies involve injecting preformed antibodies for COVID-19 into a patient who has recently acquired the virus. This is an example of blank adaptive immunity.
Let's discuss the history of vaccinations. The earliest known form of vaccinating people comes from a concept or a method of variolation. It was done so in order to vaccinate people against smallpox virus. If you look to the image at the left, you can see a patient being administered this variolation process. And what the process involved was a sick individual who had smallpox lesions. Those samples were collected from the lesions, they were dried, and they were powdered. And then they were administered. In this example, it's called nasal insufflation, where they blow that sample into the sinuses of the patient. The patient then would develop smallpox, but hopefully a much more mild form of the smallpox if otherwise acquired naturally. This method has been recorded as far back as somewhere around the 15th to 17th century in China and spread throughout the Middle East, Europe, and Africa. It is really quite incredible to think that people were able to discover this method of vaccination long before the germ theory of disease had come about, or even the understanding of the existence of viruses or pathogens. The development of modern vaccinations really began with the English physician Edward Jenner. He observed that milkmaids who contracted cowpox, which was a relative to smallpox, gained immunity to smallpox, and cowpox was a much less severe disease. He hypothesized the immunity was protective to smallpox. To test this hypothesis, he took a solution made from active cowpox legions and he injected it into a young boy who then developed a mild fever and other symptoms, but otherwise recovered and gained immunity to smallpox. Later, Louis Pasteur developed vaccines for many different illnesses, diseases such as rabies, cholera, and anthrax. Scientists have come a long way since the time of variolation, Edward Jenner, and Louis Pasteur. Today, scientists have developed adjuvants that are substances that are added to vaccines to improve the immune response to the administration of a vaccine. Aluminum salts, for example, have been used for nearly 90 years. There are newer adjuvants that are lipid-based, and there are even different compounds that we use, such as CpG, which is a synthetic nucleotide that mimics bacterial and viral nucleotides. The object of which is to try and get a really strong immune response to a artificially actively acquired vaccination, so that when you have a second exposure an incidental exposure, you will have a strong memory to that particular pathogen. The way in which the antigens are presented in vaccinations can come in different forms, one of which is called live attenuated vaccines, where an individual or patient is exposed to an active pathogen but a weakened strain of it. These types of vaccines work really well in that they stimulate the cellular immunity, or the T cells, in addition to the humoral immunity, the B cells. Since there is an active pathogen injected, they tend to not require adjuvants and require fewer, less frequent boosters, or none at all. However, you do not want to administer these types of vaccines to immunocompromised individuals for obvious reasons. They might revert back to being virulent again. Examples of live attenuated vaccines include an oral typhoid vaccine given as capsules. Next, we have inactivated vaccines, in which the whole pathogen is killed or inactivated. They cannot multiply within a patient, so they are less antigenic and require adjuvants and boosters to help elicit a stronger immune response. They only stimulate the humoral or B cell immunity because cells cannot be infected. You will not use any sort of T cell recruitment. An example of this would be the flu shot or influenza vaccines. So no, in general, the flu shot does not give you the flu. It can, however, give you flu-like symptoms. Subunit vaccines only include the most antigenic parts of a pathogen, and they typically require multiple boosters. An example of this might be H. influenzae's glycocalyx, which is used for the HIV vaccine. Toxoid vaccines are really interesting. They can be used to protect against secreted bacterial exotoxins by vaccinating with inactivated forms of those toxins. Again, since we're not dealing with an active infection of cells, we will only elicit a humoral immunity, and these often require booster vaccinations. Examples of this are the tetanus, diphtheria, pertussis toxins used in Tdap vaccinations. Then we have something called a conjugate vaccine. This is where you can take a antigen, such as a capsule, a glycocalyx, a polysaccharide, and covalently bind it to a harmless protein to make that capsule, glycocalyx, polysaccharide, whatever it might be, antigenic. That is, it appears to be an antigen to the immune system. An example of this is the Streptococcus pneumoniae vaccines called Pneumovax 23 and Prevnar 13.
Okay, that's the end of this lecture. We'll see you next time.